You're listening to BioTalk with Rich Bendis, the only podcast focused on the biohealth capital region. Each episode, we'll talk to leaders in the industry to break down the biggest topics happening today in biohealth.
Hi, this is Rich Bendis. I'm the host of BioTalk, where we interview leaders, entrepreneurs in the biohealth capital region, and today we're fortunate to have a serial entrepreneur with us who's done it more than once in this area, and it's Bill Hearl, who's the CEO and founder of Immunomic Therapuetics. Bill, welcome to BioTalk.
Thanks, Rich. Happy to be here.
Great. We're going to tell all of the listeners about all of your history, the ups, the downs, and basically more about the successes and what the future looks like for ITI. So really been looking forward to this interview, Bill.
Should be fun. And they're actually, over the time, I think there's been a lot more horror stories.
Well, you know, it's important for emerging entrepreneurs to understand everything doesn't go as planned.
Absolutely true.
And we probably have both had some of those things that haven't gone as planned. So we'll touch on that. We'll talk a little bit more about what to do rather than what not to do. So why don't we start first by giving a little bit about your background, history, so everybody understands where you come from to get to where you are today.
Okay, that'd be great. I'm a Ph.D. biochemist, and I got my degree at Oak Ridge National Laboratory through the University of Tennessee. And when I actually moved to Maryland straight out of graduate school and had a postdoc at USUS, and very quickly realized I wanted to work in the biotech field in a company. And so my first employer was Electronucleonics, which for the old timers know that was a company that was at the forefront of HIV blood testing research. So I really moved pretty quickly into immunology and immunology testing after working all my career in biochemistry. And that was a pretty significant shift in thinking for me, and it got me into a lot of product development early on. So as a bench scientist, I was very fortunate to be exposed to a lot of different elements in a relatively significant company in our space, and had access to a couple upper layers of management that were very instructive to me. So for people that are looking at it from a scientific perspective and wanting to be an entrepreneur, I think diversification is kind of a nice thing to build into your career as early as possible. I worked at Life Technologies, had a cup of coffee at iGen, then went to Kierkegaard & Perry Labs, or KPL, also in Gaithersburg. And at KPL, I was able to take over an R&D operation. And that's really where we started building the foundations for being an entrepreneur. The group started to develop a number of products, and we built some critical mass around something that I thought could become a company as a spin-out company. And so we formed a company called Capital Genomics in 2000, which was a combination of genomic discovery and antibody development. And so the idea there was to identify novel gene transcripts and turn them immediately into antibodies looking for druggable targets. So that one really took a couple of strange turns. We ended up buying Dynex from Thermo and turning that into a success for the company. But that really wasn't how we planned on exiting Capital Genomics.
Well, that's pretty typical with most companies. They don't end up the way they started.
As we were winding that up, the investment banker said to me, "Hey, Bill, if you could get into vaccines, I'd do it." And really that became the genesis of the search for the technology that would form Immunomic Therapuetics.
And then that search took you to a university, I think, didn't it?
Yes.
Yeah.
Tom August, who is a distinguished professor at Johns Hopkins. Tom is, I think, 92 now, and still does active research, which is really remarkable. And that way, he's got a 70-year research career. Really phenomenal. And so Tom and I were talking over breakfast about things that were going on in his laboratory in the vaccine space, and that led to a discussion around licensing the LAMP platform out of Johns Hopkins and making what became Immunomic Therapeutics.
And can you explain what LAMP is, please?
Sure. LAMP is an abbreviation for a gene, lysosomal associated membrane protein.
Everybody will remember that.
Yes. It's much easier to say LAMP. And so the LAMP gene itself is highly conserved in nature. So any animal that has an immune system has LAMP. Chickens have LAMP, fish have LAMP. Obviously, humans have it, monkeys, dogs, mice. And there's a very high degree of conservation of the key gene sequences in LAMP. So actually, human LAMP works perfectly well in mice. And so what Tom observed was that this gene, when the protein is expressed, localizes in immune cells in the compartment that has protein that's responsible for teaching the immune system about how to recognize foreign antigens. And so Tom kind of figured out, hey, if I stick a protein sequence in the middle of LAMP, logically it's going to end up in the same compartment. And so that turned out to be true. For myself, I had been working in nucleic acid immunizations at the time for about 15 years. And Tom's observation was an eye-opener for me. I realized that, aha, this is kind of the missing link that we've been looking for in the vaccine space, which is a way to directly communicate with professional antigen-presenting cells. That was the crux of the technology.
And then basically, I would assume you licensed that technology from Johns Hopkins, correct?
Yes.
How did that process go?
Well, it was interesting, that's for sure. It took several months. I had the support of Tom behind me very strongly. We were able to work out an arrangement with the tech transfer office, which at the time was evolving. And then I think Wes Blakesley came in, and Wes really helped me get the company going. So once we got over the hurdle of getting the initial license, they were very instructive and helpful in keeping the license in ITI and working with us when times were lean, especially in 2009, and allowed us to continue to develop our technology and the platform even when the company was a little cash-starved.
So what year did you actually form ITI initially?
That was in 2006.
In 2006. Okay. So you had met Tom many years before that, correct?
Yeah. Tom was on the scientific advisory board of Capital Genomics.
I see. Okay.
I had known him since about 2002 or so.
And then you mentioned there were some dry times.
Yes.
So, I mean, everything just doesn't go up a straight curve when you're doing an entrepreneurial startup. So talk a little bit about the startup, how you got going, and then the dry time, and then how you overcame the dry time.
The company, when we started it, we were fortunate that we had a license in our back pocket with the Geron Corporation. And Geron had used LAMP in a clinical study in cancer patients at Duke and had actually approached Johns Hopkins about licensing LAMP for that particular application. So I convinced the tech transfer office that I could get a better deal than what they had on the table, and we'd split the difference. So we got the deal done, and that's actually what we ended up doing. They made more money, and I got seed money to start Immunomic Therapuetics. We got $75,000 from TEDCO. That was really helpful. And then at the time, the Montgomery County Economic Development Office was giving out $50,000 loans, and we got that. And so that put together the capital that got the company off the ground. Tom had been working with a group in Brazil on HIV. And if you kind of go back and looked at Tom's publications early on, particularly with LAMP, he was doing a lot using the LAMP linked to different HIV proteins as a therapeutic treatment. So we started a collaboration with a group in Brazil to conduct a clinical study for a therapeutic application of LAMP. And as things kind of went along, things were great at the beginning, as often happens with those projects. But as it became evident that the project was going to cost more than the sponsor had originally envisioned, they decided they didn't want to do that anymore. So they kind of left me high and dry in January of 2009. We had believed that we were going to be able to fund the company, I think, through 2009 off of this HIV project before we were going to be able to shift into the things that we really wanted to work on. That made it so that Immunomic Therapeutics moved to my garage for a few months.
Rather than starting in your garage,
yes,
you went back to your garage.
Went back to the garage.
Okay.
Perhaps when I started the company, I had envisioned the fact that lean times were possible. And while I wasn't forecasting any sort of biotech bubble, when one happened, that preparedness paid off because we didn't have to close the company. We had no debt. We didn't owe anybody any money. We were able to work with Johns Hopkins to maintain the license while we were out raising money. And I'd gotten a commitment about the middle of the year from the Life Science Greenhouse, Central Pennsylvania, to invest in Immunomic Therapeutics. That allowed me to get a group of angel investors behind that would do matching contributions. So it took a little while to get through the due diligence. And so when we got to the end of 2009, we were almost out of money. And I like to say this, I called up Bernie Rudnick, who was my acting chief financial officer at the time, and I said, hey, Bernie, the bank balance has 3 digits, and 2 of them are after the decimal point.
Right.
and so I said, help. And so who turned out to be my largest investor, stepped in and supported us and bridged us to the first allocation of funds from the greenhouse, and then we got rolling from there. We had another family fund that came in and invested an equal amount of money, and by early in 2010, we had accumulated enough mass to really get the company up and running again and focusing on our allergy products. About that point in time, we also were starting to focus in on a clinical study that was going to use the Japanese red cedar, which I'm sure we're going to come to and talk about. But that allowed me to apply for some funds from, at that point in time, there was a program that the government had came up with to try to restart businesses, which was the qualifying tax credit. And so we took advantage of that and got another $250,000 through the IRS. And in that case, the IRS was our friends, and they certainly helped us. And so all those pieces kind of came together and got us going and got the company off the ground.
So that's what they call bootstrapping. And also an entrepreneur following the money, which meant that even though you're from Montgomery County, Maryland, this required you going to central Pennsylvania to get some funding to enable you to continue the growth of your company.
That's correct. And I am infinitely grateful to the group in Harrisburg. I mean, they were very supportive, and we were happy to have, we first had a facility and offices in Lancaster and then moved to Hershey. And at Hershey, we built our first process development laboratory so we could start evaluating manufacturing product. And so we had a good relationship, still do, of course, with the greenhouse. And I'm a big fan of theirs, too.
You also brought up Japanese red cedar. I guess if we talk about the evolution of ITI, and then you're getting into the Japanese red cedar area, it led to really the ultimate success for ITI. So let's talk a little bit about that evolution and then where it took you.
It's kind of interesting, and I, as I mentioned, I was meeting last night with my colleague from the University of Florida, Dwayne Mitchell, who is running our glioblastoma program. And Dwayne had actually worked in the laboratory of Ellie Gilboa at Duke, which was one of the things that really attracted me to the LAMP platform. Gilboa had done a study with human telomerase in prostate cancer patients and had showed an amazing response with that vaccine when it was linked to LAMP, and he did a very nice study. And so I actually had asked Tom, hey, why aren't you working on cancer? And he's like, I looked at the paper publication, said maybe we should be doing that. In my mind, I always wanted to come back to it, but it was very difficult in my mind to raise enough money to support cancer clinical studies. So we were looking for something as an alternative, and a lot of people naturally were drawn to influenza, but I felt like influenza was a field that was unlikely to be successful because of the challenges of the seasonal nature of the disease, as well as the number of competitors in the space. And so coincidentally, there was some work being done by a collaborator at the University of Singapore, and she had shown that LAMP actually reversed allergy in mice. And so once I saw that, I was like, yes, that's exactly the right application. It was the perfect storm for our company, which was an opportunity for a very large unmet medical need, a perfect application of the technology, and a huge market. And so we really
exactly what investors like, right?
Absolutely.
Yeah.
Unfortunately, nobody ever heard of Japanese red cedar, and one of the questions always comes like, well, how did you pick that? The natural inclination when you start working on allergies, well, let's do ragweed. Another one that people would kind of attract to is cat, which is a whole nother story. But we were looking at these seasonal allergies, and we realized that ragweed was a problem because if you're doing an outdoor study, people that are allergic to ragweed are at the same time that ragweed is blooming, so is every other weed and grass. So how do you distinguish in a clinical study ragweed from the effects of grasses and other weeds? As it happens, Japanese red cedar, it blooms in February. And so at the time that those cedar trees are blooming, it's pretty much the only game in town. And in Japan, it really is the only game in town. After World War II, Japan needed to plant trees to prevent erosion and so on. And so somebody decided to plant millions of acres of Japanese red cedar trees. And interestingly, they've become a very popular ornamental tree around here. I see them all over the place. People don't realize that they're making this allergy bomb in their front yard, but so be it, right? The trees are absolutely beautiful. They're fast-growing trees. They grew up to about 35, 40 feet tall, and each tree makes 2 to 4 kilos of pollen. And so if you can imagine that a 50 conical tube holds about...
That's annually.
Right, annually.
Annually.
A 50 conical tube holds about 5 grams of pollen. Think about the volume that you're looking at for kilos of pollen. So as these trees matured, these giant clouds of pollen started coming off the trees, and people originally thought they were forest fires, but they were just giant pollen clouds. And so right now, 35 million people in Japan have cedar allergies. So we looked at it as a unique opportunity where the allergen was very well defined, and it was something that was easy for us to build a vaccine and do a clinical study. So we chose Japanese red cedar. And as I was going out doing pitches, people didn't understand necessarily that 35 million Japanese translated into a $70 billion market opportunity. And it was an unfamiliar concept, but they did understand peanut. So we created a peanut vaccine to run in parallel. So we were developing those two allergy products in parallel. And what we did with the Japanese red cedar, it was such a simple thing. We wanted to just do a safety study. And so I don't know if you're familiar with skin testing, right?
Yes.
We do for allergy testing. At the clinical site, skin testing was the criteria we were using for admitting people to the study. And the gentleman from the clinical site called and said, hey, some of these people have welts from the testing about the size of a $50. And so I said, maybe we should test them when they're finished and see if it changes. So they went through the protocol, they got their dosing, and the first 6 patients came in for their closeout testing, and they called back, and they came in and told me all 6 of the people were skin test negative. And I said, well, what's wrong with the reagent? There must be bad. There's no way that the skin test could go away. And so we confirmed that the skin test was valid, and at the end of the first study, 14 of the 18 people were skin test converted, which is an unprecedented response to allergy. Bruce Mackler, who was my head of regulatory at the time, Bruce immediately recognized that opportunity, and we went ahead and got the people to come back in, take a booster shot, and then at the end of the boost period, we had 100% conversion. We used that to market that back into Japan and took that to the regulatory agent in Japan, PMDA, as well as we presented it to 20 different companies in Japan and found a partner for Japanese red cedar, which turned out to be Astellas Pharma.
How had the company progressed at that point? How many people did you have in it? And at what stage of development were you when you started to look for your strategic partner?
At the time that we started looking for the partner, I'd say we probably had 10 employees. And so we had kept the company very lean. We maintained the number of people that we needed to do the R&D work, and then we partnered out or contracted with different consultants to contribute the elements that we needed to do the clinical studies or outside manufacturing and quality assurance. So all the components that we needed, we bought as we needed and didn't rush out and hire a bunch of people. And we just maintained a small footprint in the building that we're in today here on Brochart Road. That was what was sufficient.
It's nice that you came back to Maryland. We'll talk about
Yes.
that.
So that worked out really well for us in terms of the size of the company, and we had just the right combination of skills. And I've really been very fortunate that everybody that I've hired for Immunomic Therapuetics has been a superstar. You know, how often does that happen? But my co-founder, Terry Hyland, who's ran the R&D, Terry and I have worked together now for over 25 years, getting all the way back to when we were at KPL. And Terry's got a very shrewd eye for science and really did a great job at zeroing in on the technical issues that were going to be necessary to be successful and how to analyze the data coming back from the clinical study, kind of blood tests we needed to validate the results. So what we really had going on was that I was out raising money and beating the bushes for our business development partner, and Terry did everything based
Mr. Inside and Mr. Outside.
Right. It was a great partnership and still is, and it's a structure that works for me.
And when you met Astellas, tell me a little bit about the evolution of that relationship and how long it took to culminate into the successful exit that you had with them.
That was a surprisingly speedy deal. We actually did our road show into Tokyo in February of 2014. We sent out 20 invitations and had 11 meetings. 2 of them turned out to be with competitors, so we discarded those. So we had 9, and then we got 6 into due diligence. Now, what was very interesting is that all the companies that entered due diligence told us that PMDA would never approve a nucleic acid vaccine and that it was an impossibility, but that was sort of like waving a red flag in front of a bull for Bruce. And so Bruce initiated a program to get us in front of PMDA very quickly. It's kind of interesting because actually, I'm pretty sure Astellas knew what we were up against. And I almost wonder if they were testing us to see if we could figure out how to navigate the regulatory waters in Japan before they made us an offer.
Right.
So we went to PMDA and made our presentation, and they told us that they would be willing to accept what they call a CTN, which is their version of an IND.
IND, okay.
And they would accept our data that we had collected in Hawaii in native Japanese patients as valid. And so we went back over to Astellas right after that meeting. We went right over there, and we said we had this data. The head of the clinic looked at me and he says, that is not possible. They did not say that to you. And I said, yes, they did. And while we were on the train going back to the airport to go home, they called me and said, don't send them anything. We want to do this.
Don't send who anything?
Don't send PMDA.
Oh, okay.
Don't send PMDA any information
right.
now. We want to do this deal. That was actually October of 2014. And so I just told them, well, we don't have a deal. If you want a deal, we got to move quickly, or we're going to file by the end of the year. We went back in November, had the structure of a license, and then signed that deal by the end of January. So that was the $15 million up front for Japanese red cedar with $55 million in license milestones and an additional royalty.
And so that's really an 11-month time period from the time you first came in contact with them to where you consummated this transaction.
Correct.
And then after you got this deal done in 2015, in January, what was the progression from that point further with Astellas?
Well, as soon as we finished that deal, I issued a mandate to my team that we were going to do everything we could to demonstrate our commitment to being successful with the Japanese red cedar program. Earlier in January, I had hired Tim Coleman as our head of operations, and Tim had to come in and take over making the product for them for the next study that they were going to do in Japan. So we put together a CMC team so we could make product. We really dedicated our R&D team to addressing that, and we had our clinical group start interacting directly with Astellas. So across the board, the Astellas program was our top priority, and we were singularly focused on making that successful. And it left a favorable impression. And they had told us after the first couple meetings that they planned on coming back to Immunomic Therapuetics and making a proposal at the end of the summer about perhaps acquiring some other products. So sometime in late July, they approached us about buying the peanut. They made us an offer on that. We looked at it and thought it was not a bad offer, but if I was supposed to sell them peanut, how could I sell any other food allergens? So I said, if you want to buy peanut, you have to buy all the food. Well, we also want house dust mite. Well, if you want house dust mite, you have to get all of environmental. So then we're talking about the whole ball of wax.
Right, the whole company.
That whole segment of business. Then they made a proposal to my chairman on the side, and he said, well, you can give Bill that number, but he'll probably be insulted.
So it's good guy, bad guy.
Right. And so they came back with a better number, and I thought the number was good, except it was just missing a royalty. And so we ended up settling on the $300 million flat and a 10% royalty flat, and they acquired all the rights to allergy. That was a life-changing moment for certain.
Well, I'm sure it was. And when we talk about the $300 million and the 10%, the $300 million was at closing, I would assume, right?
Yes.
A one-time deal.
Yes.
And the 10% royalty, is that in perpetuity or is there a time scale to that?
It's a 10-year from first sale.
From first sale. Okay, great.
Yeah. Actually, that's one of my fond memories of business is Eric Windsor, our CFO, came into my office and says, hey, Bill, you need to open up the Bank of America account. And there was a number across the screen as wide as the screen with zeros. And I was like, yes, that is.
And they were on the right side of the decimal point this time versus the other side.
That's exactly it.
Congratulations. That is a great success story, and I'm sure everybody loves to hear about those kind of successes. But you're not done yet. You did the deal. You sold the allergy component of it, but ITI still existed. And what did it have in the company after you had made the sale to Astellas?
What's interesting about it is that when we started the company, as I mentioned earlier, like the cancer component was really what I thought was the home run for the company. And the allergy activity was really more a validation process and a stepping stone to what I really wanted to do with the company in terms of immuno-oncology and cancer vaccines. So while we were doing the allergy work, we had developed relationships with a couple of academic laboratories that were actually using the LAMP technology in a couple clinical settings. And in particular, the one that got the most attention from us was the work that was being done by John Sampson, who's at Duke, and Dwayne Mitchell, who's now at Florida, with glioblastoma. I mean, everybody familiar with the John McCain story knows that 14 months, I told my wife, unfortunately, I said, if we can't get Senator McCain into our study, 14 months from now, it's not going to be good. Right. Almost to the day, I think.
Came true.
Kind of the way it worked out. Well, the study that was originally shown to me by Dr. Sampson showed that about 50% long-term survival in glioblastoma patients that were able to be treated very early, as soon as the initial diagnosis of GBM, if they came in and got into the program, they had a 50-50 shot of long-term survival. And we're talking about years. Right. These papers are published in Nature, and it's really very exciting and very compelling data. The only limitation is for that kind of study and for that kind of work, the patient population is too small to be statistically significant. So while that was going on, we just tracked that, and we were actually making a commitment to help support the clinical study with a gift to the University of Florida to help support Dwayne's lab in developing this. And then very fortunately for us, the company that had been in line to license the whole vaccine dropped out, and we moved in very quickly and opportunistically, I would say, took control of the vaccine formulation for the glioblastoma. So we own all the IP around that now, and we're fully supporting and are supported by, it's a mutual relationship between Dwayne Mitchell and John Sampson and Immunomic Therapuetics to develop this. So that is our lead candidate. And we have several other formulations that are all designed around using our platform to attain a global immune response against specific cancer antigens, and in particular cancer antigens that are related to virally mediated cancers. So in glioblastoma, the problem that they have is that most of the tumors have what's believed to be a secondary infection of cytomegalovirus. So CMV is there, and the proteins are there, and it makes it a very good candidate for immunotherapy because you can see it, and the immune system can see it. So you get a good response rate in quite a large percentage of the population of patients.
How long do you see the current research that you're doing right now before you're going to get into an IND or progress significantly into the regulatory environment?
The study that's going on now is a phase 2.
Okay, it's a phase 2.
So that one is intended to recruit 160 patients into 3 arms. It's a blinded study with a placebo group. So it's a well-designed, statistically powered study, which when we get to the point where we can take a look at it, if it's successful, I would imagine that we would move very quickly towards a conversion to a phase 3 and compassionate use applications. Immunomic Therapeutics on our part is filing an IND in the next few months to create the commercial version of that, to get it out of the academic laboratory and into a commercial setting and prove that we can replicate their results as a commercial entity. So that's our main plan with that.
And I would imagine the capitalization and funding for the second generation of ITI is a little different than the first time you started.
Yes, and it's kind of an odd situation, I'll be honest with you. I mean, we started out, funded the company primarily through angel investment, and we did not really take any institutional money except from the greenhouse, and then we took some family office money. And so once we did these different deals that we've had quite a bit of non-dilutive capital come in. And of course, we rewarded our shareholders and we kept the money in the company, but I actually haven't had to go out and raise money for almost 4 years now. So looking at the cancer activity, of course, now you're looking at a whole different scenario. And so we want to go out and raise a Series B of around $30 million to accelerate the different cancer programs. We have 2 or 3 things that are really very exciting in immunotherapy and immuno-oncology. And we see that as a stepping stone. The company's ready to position itself for potentially public markets. And so I think that's the right path for us, is to bring in the right type of institutional investment that can help us with strengthening our board with some industry leaders and putting us in the right spot to really move this cancer program much, much quicker. It's time.
One of the nice things for this region is you've made the decision to do this in Montgomery County, Maryland. I'd like to get your impressions of how it's progressed today versus when you first started back in 2000 up to 2006. If you look at this ecosystem and what you're doing today to build ITI versus where you were 10 years ago, how have you seen the progression?
Early on, when we were out looking for money, I was very frustrated with the setting that we had in Maryland and this region in general. There was a couple of players who were making some investments, but it was really pretty limited. And I don't want to say it was a closed shop, but it was sort of like if you weren't an HGS guy, your chances
if you
of
weren't
getting
in the
money,
club,
right? If you weren't in the club, you weren't getting the money.
Right.
And I found the opportunities. I raised a lot of money through the different Keiretsu forums, and actually did very well in the Pacific Northwest, in San Francisco, and in Philadelphia, and also at Fisher Island in Florida. I mean, we went on the circuit, and we did really well. But the environment in the Philadelphia, Pennsylvania area was much more open to in making these kind of investments in biotech company than what I was seeing in Maryland. So it fit for us. Now I really think that we're starting to see some things moving in a positive way. For example, as you know, BHI has taken a real lead in driving the events that are going on that have been hosted at MedImmune the last couple of years that have just been fantastic events. And it's really attracting attention to the biotech community that's here. I mean, after all, you have a tremendous resource pool between the NIH and Johns Hopkins, University of Maryland system, Georgetown. The region is loaded with talent and loaded with good ideas. Somehow or other, we just haven't really, until now, been able to figure out how to start to extract that talent into viable companies. So things are finally starting to move, and there are people making investments, and there are events, and we're getting the right kind of attention. So you don't have to be in Boston, and you don't have to be in San Francisco or San Diego to be getting biotech money.
I love to hear that, Bill. There's no sense saying, though, that unless you believe it, unless you're actually seeing the evidence of that happening around here. And as you know, we're going to have the first BioHealth Capital Investment Conference in October 9th and 10th. And we now have 40 committed investors who are going to come into Maryland to look at 85 companies and do one-on-one meetings with them.
I'm thrilled about that. I think that's exactly the thing that's been needed. I think for the first time in quite a while, we see kind of renewed energy around, of course, what you're doing at BHI, but I think also the Maryland Tech Council, the bio arm of that, really finally seems to be picking up some steam. And it's been a while since, kind of harkens back to the old days of MD Bio, when it was a trailblazing organization. And I think we kind of lost our mojo somewhere along the way. And now I think we're starting to get it back.
Well, maybe that resurgence has something to do with you, because I think you're on the board of both of those organizations now, aren't you?
Maybe.
Yeah, both Maryland Tech Council and BioHealth Innovation. So
that could be true.
I thank you for your support for BHI, by the way.
Yes.
So let's talk about a little bit. You've been very successful, and I know one of the things you like to do is to sort of give back. So you're trying to pay it forward in a way. Talk a little bit about what your goals are and how you're going to give back to keep this ecosystem growing.
One of the things that made Immunomic Therapuetics possible was just the ability to get a lab bench and be able to do some experiments without having to pay a huge amount of rent, and just enough to get enough so that we had some critical mass of data to augment our presentations. I have never forgotten the value of that. We actually had a laboratory inside Expression Pathologies over on Medical Center Drive, and it was a bench in a closet, but who cares? That worked for us. And now that we've expanded here to our facility on Brochart Road, we have some nice extra space. And last year we ran a contest. We took in one company to let them benefit from having a lab bench. And now we've moved on to a second company, I believe it's Dave Krisman's company, KRI Technologies, is going to have a lab bench here in the immuno-oncology space. So we're kind of enthusiastic about that. And we're even looking at potential academic collaborators perhaps taking a room from time to time and using that. And we've done this in the past as really kind of like a no strings attached, but it's designed in a way to be synergistic with our efforts and to engage our scientists as well as to give the company a chance to get off the ground. And frankly, having done as many stupid things as any entrepreneur can do and still be standing, I don't see any reason for anybody else to have to reinvent the wheel.
Well, I mean, the key and the benefit to one of these entrepreneurs coming in and getting a bench here, just think about having the ability to get a mentorship from you as well, even if it's once a month sitting down to have a lunch, but also the interaction with your scientist on a day-to-day basis, if needed. Walking around in a successful company is got to be great for an entrepreneur.
There are resources here that people probably aren't even thinking that they would really need to have a conversation about. But if you're intending to do a regulated product, you should be thinking about what kind of filing you're going to be making at the FDA before you do experiment 1. Because all that data is going to be compiled into some sort of package that you're going to deliver to the FDA sometime in the future. Why not get it right the first time?
I think the key there is that it's not a linear process. And the other thing is, if you have the astute knowledge of people who have been there and done that, you can eliminate some of these roadblocks that entrepreneurs are going to naturally come across. But if you know they're coming, you're going to know how to address them better than if you just get this blind wall in front of you.
That's correct.
And Bill, I respect what you're trying to do to help build the entrepreneurs. Let's talk a little bit in closing here about your personal and corporate goals for the future.
Personally, I'd like to have a single-digit handicap.
So, how close are you?
I'm getting there.
Okay.
I've improved. I'm better than I was, but I try to schedule my massive amount of travel during non-golfing months.
Oh, gotcha. Okay.
And for the company.
But you're not called for the Ryder Cup team this year yet, though?
No, and not likely anytime soon
Okay.
either. But for the company, I think what we'd like to do is we're very fortunate that we've been able to put our investors in the black. I mean, there's not that many biotech companies kind of look back and say, well, everybody's operating on the right side of the ledger. And now we want to make sure that we continue to create value, but in a very specific way. I'm very passionate about the idea of finding solutions for cancer patients. And I think cancer patients face a tremendous uphill battle just from having their diagnosis, not knowing what to do, and being lost. And because of that is why I participate on the board of Hope Connections, for example. There needs to be a connection between our community that's trying to develop drugs to treat cancer and the cancer patients themselves. You can't do a clinical study without cancer patients, and cancer patients are often lost in the information world and don't know where to go. So I have a philosophy, just terms for the company, that if you find a solution for cancer, you don't have to go looking for money. The money will find you. And so we focus on trying to find a solution and not being overly burdened with the idea that how that's going to translate into value. It will. I mean, if we can show conclusively that we're improving the life of glioblastoma patients, you can't tell me the company's not more valuable and significantly more valuable. And so it's the same is true for the other targets that we have in our pipeline. So I think that we've got a great team. I want to continue to develop the company and give people a chance to grow into their careers. You know, I haven't forgot what it's like. Hopefully, I haven't forgot what it's like to be like a 30-year-old scientist that wants to do something and conquer the world, kind of thing. And we give people the freedom and the environment where they can be successful as scientists and at the same time advance the corporate objectives. And I'm fortunate that my board and the shareholders have been really supportive of that concept, and they're excited about what we see as a future for Immunomic Therapuetics as one of the companies that's going to make a dent in treating cancer patients.
I think those are admirable goals, both of them. You need to enjoy your personal life as well as what you're doing corporately. And having two parents who both had cancer, and almost every family in the United States is touched directly or indirectly by cancer, which is just unfortunate. But we wish you the best of luck and the success for everything you're doing here at Immunomic Therapeutics. And last thing I'll close on is any words of wisdom you have for entrepreneurs who are embarking or in the middle of the process going through what you've been through successfully.
Yeah, I think that my word of advice is if you're going to do it, go all in. There is no half measures in this sort of thing. Either you're in or you're out. And you got to make total commitment. It's a big sacrifice. You should know when you're going into it that if you have black hair, that's not going to last very long. And that's the only way to be successful, is to be totally committed to your mission, and you have to have a singular focus for success.
Great words of wisdom. I echo exactly what you're just saying, Bill. I thank you very much for what you're doing for the BioHealth Capital Region and what you're doing for your company and the culture you're creating. Also for supporting biohealth innovation with your support of our board. So thank you very much for being on BioTalk. I'd love to come back and follow up with you as you continue the progress of this company.
That sounds great, Rich. It's my pleasure.
Thanks for listening to BioTalk with Rich Bendis.